Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 5th February 2021. As all of us know, we are into the revision mode and we are revising for the third round in the form of CBSE sample papers. As I promised you, I am going to give you around 10 to 11 CBSE sample papers. And I have also requested you to keep a notebook separate. Write whatever I am giving to you in that notebook. It will be very handy during your exams time. And yesterday's session we covered up to question number 26th. Now let us finish this sample paper today, and let's go straight to question number 27. Of course, section C, all of you know, has got five questions. Each question has got 3 marks. So 5 3s are 15 marks. And section D also reverse 3 questions with 5 marks each. 3 into 5, 15. So section C 15 marks. Section D 15 marks. So 30 marks we are going to cover up today. Question number 27. Would it be appropriate to use DNA probes such as VNTR in DNA fingerprinting of a bacteriophage? Now the answer. What is VNTR? Normally we will ask you sometimes 1 mark question also. Variable number of tandem repeats. That's the expansion of VNTR. Is a location in the genome, the group of genes, where a short nucleoside is organized as tandem repeat. Analysis of VNTR is used for many purposes, including DNA fingerprinting. But bacteriophage does not have too many DNAs. Rather, only a few strands of DNA are available in bacteriophage. This does not leave scope for repeating sequences in DNA. Hence, VNTR cannot be used in DNA fingerprinting of a bacteriophage. Question number 28. Name and explain the 2 types of immune responses in humans. There are 2 immune responses. First one is the primary immune response. This occurs when an antigen, of course antigen is a protein, comes in contact with the immune system for the first time. During this time, the immune system has to learn to recognize the antigen and how to make an antibody against it. Anti against the body. Which body? Antigen. And eventually produce memory lymphocytes. Whereas second one is the secondary immune response occurs when the second time or third, fourth, whatever it is, the person is exposed to same antigen. At this point, immunological memory has been established and the immune system can start making antibodies immediately. It is based on memory. Next, this is how we are seeing antibodies in the blood. So immunological memory, and this is the primary response, first exposure. Whereas second time you see it's very fast, and mainly IgG and IgV, these are responsible for making the memory-based antibodies. Question number 29. Explain the process of splicing, capping and tailing which occur during transcription in eukaryotes. I said all these 3 are technical words. Biotechnology. The process of transcription, molecular biology. The process of transcription in eukaryotes, eu means clear, karyon means nucleus, can be explained as follows. Splicing. The primary transcript of RNA undergoes splicing, by which the introns are removed and the exons are joined together. So there are 2 things, exons and introns. So if exons are joined together by removing intron, that is called splicing. Next, capping. Methyl guanosine triphosphate is added to the 5' end of hnRNA in capping. So capping is addition. Tailing adenylate residues about 200 to 300 are added at the 3' end of hnRNA in tailing. Now as you are seeing here, you can see this capping, then also the introns, exons. Now introns are removed and entrons, and then you can see the presence of mRNA, and then you can also see the formation of the messenger RNA. So this is basically a transcription, eukaryotic transcription. Question 30. Pollen banks are playing a very important role in promoting plant breeding program the world over. How are pollens preserved in the pollen banks? Explain. How are such banks benefiting our farmers? Right? Any two ways. So we talked about pollen banks. Just like we have got State Bank of India for our cash, we can deposit pollens which are high quality ones. So pollen banks are used to store pollens for a very long period of time in a viable conditions. Pollens are preserved in a bank using cryopreservation, that is storing at low temperatures. They are stored in a viable condition in low temperatures. How much? Minus 196 degrees Celsius using liquid nitrogen. The important applications of pollen banks for our farmers are the following. 1. To preserve the agricultural biodiversity in the form of preservation of valuable genetic resources. We are protecting the valuable genome. Second, these pollens can be used in various crop hybridization breeding programs, biochemical and physiochemical studies such as the study of allergens. Okay, and you have got a choice for this question. What is the choice? Rita and her parents were watching a TV serial in the evening. During a commercial break, an advertisement flashed on the screen which was promoting the use of sanitary napkins. Rita was still watching the TV. The parents got embarrassed and changed the channel. Rita objected to her parents' behavior and explained the need for these advertisements. Question 1. What values did the parents show? The parents were traditional but understood the need for such advertisements. They showed maturity and openness later. Question 2. Briefly describe the phases of MC, menstrual cycle. So draw the diagram like as it is drawn and write the 3 phases. Since it is a 3-mark question, only you have to mention. Okay, that is phases, details of different ways describing the diagram. Menstrual phase, then proliferative phase and secretory phase. 3 phases. Briefly you explain what is menstrual phase, what is proliferative phase, and also out of the 28-day cycle, how many days this will happen, how many days proliferation, how many days secretory phase will happen, and what is the influence of hormones. That also you have to make it. Okay, that you need to talk every stage. Next, section D. Question. These are, as I said, it's a 5-mark questions, and there will be internal choice for each one. So you can attempt whichever is having 1, 2, 3, 4, 5, so that probability of you getting more marks is more than attempting a straight question. Okay, where lot of description is there. Question 31. Given below is the TS of human ovary. Identify the following diagram: corpus luteum, secondary oocyte, antrum, primary follicle and primary oocyte. So there is a diagram TS of ovary. So one has to identify what is this A, B, C, D, E, F. So A is Graafian follicle, B is antrum, C is secondary oocyte, D is corpus luteum, and F is primary follicle and primary oocyte. Question 2. Explain the changes the primary oocyte undergoes while in different follicular stages before ovulation. In human females, primary oocytes are formed during the embryonic developmental stages in the fetal ovaries. Primary oocytes start dividing and enter prophase 1 of meiosis to remain suspended at this stage. Each primary oocyte is surrounded by a layer of granulosa cells and becomes the primary follicle. C. The primary follicle, when surrounded by more layers of granulosa cells, is called a secondary follicle. Now there is a choice for this. Before that, still more, secondary follicle transforms into a tertiary follicle with the development of fluid-filled cavity. We call it antrum around the primary oocyte. Granulosa cells become organized into outer theca externa, inner theca interna. And now primary oocyte completes meiosis 1 and forms a larger haploid secondary oocyte and a tiny first polar body. You got a choice for this question. And the choice is study the illustration given below and identify A. Okay. Name and state the function of C. Identify D. Explain the role of hormones in the formation and release of A. Draw the diagram of B separately and label the parts that helps its entry into A, that carry genetic materials that helps in its movement. Now, it is again diagram based question. One is human ovum. Second one, zona pellucida. When one sperm penetrates into ovum, the zona pellucida makes it impervious to other sperm. Cells of corona radiate layer. Fourth one, GnRH, gonadotrophic releasing hormone of hypothalamus, stimulates the anterior pituitary to secrete FSH, follicular stimulating hormone, and LH, luteinizing hormone. FSH stimulates the formation of ovum by stimulating the growth of ovarian follicles. By the action of LH, the oocyte completes the first meiotic division and becomes secondary oocyte and finally release from the follicle. And the diagram is sperm. Label it acrosome, head, nucleus, mitochondria, middle piece and tail. Question 32. Write the palindromic nucleotide sequence for the following DNA segment. He has given this 5' GAATTC 3' palindrome reverse. So 3' CTTAAG 5'. I said Malayalam Gadag. Okay. Name the restriction endonuclease that recognizes this sequence. Restriction endonuclease is ECO-R1 that recognizes the palindromic sequences. How are sticky ends produced? Mention their role. Sticky ends on DNA are formed by the action of enzymes, restriction endonucleases. These enzymes cut the strand of DNA a little away from the center of the palindrome sequence between the same two phases on both the strands. This results in single strand stretches on both the complementary strands at their ends. These overhanging stretches are called sticky ends as they form hydrogen bonds with the complementary base pair sequences. And what is the role of them? These sticky ends produced from hydrogen bonds with their complementary cut counterparts. This stickiness of the ends facilitate the action of the enzyme DNA ligase. And choice, if desired gene is identified in an organism for some experiments, explain the process for the following. 1. Cutting this desired gene at a specific location. Cutting of the desired gene at a specific location is done by incubating DNA with a specific restriction endonuclease. Restriction endonucleases recognize a particular palindromic nucleotide sequence and cut the DNA at the site. Question 2. Synthesis of multiple copies of the desired gene. Synthesis, of course, multiple copies of the desired gene is carried out by PCR, polymerase chain reaction. Amplification of recombinant DNA gene is done using polymerase chain reaction. It is carried out in the following steps. Of course, we know it: denaturation, annealing, and extension. The double-strand DNA is denatured by applying high temperature of 95 degrees for 15 seconds. Each separated strand acts as a template. Annealing. Two sets of primers are added which anneal to the third end of each separate strand. And extension. DNA polymerase extends the primers by adding nucleotides. Complementarity to the template provided in the reaction. Tuck polymerase is used in this reaction, which can tolerate heat. That is the purpose why we use tuck polymerase. All these steps are repeated many times to get several copies of the desired DNA. We come to the last question. Explain the process of sewage water treatment before it can be discharged into natural water bodies. Why this treatment is essential. Primary treatment of sewage involves the physical removal of large and small particles from sewage through filtration and sedimentation. The steps involved: floating debris is removed by sequential filtration by passing through wire mesh screen. This is physical. Second, after this, the grit, soil and small pebbles are removed by sedimentation in settling tanks. The sediment is called primary sludge and the supernatant forms the primary effluent. Third, the effluent is then taken for the secondary treatment. The secondary treatment of sewage is also called biological treatment because in this treatment, sewage is subjected to biodegradation. It means that it involves the participation of microbes. The process of secondary treatment involves the following steps. So primary effluent, then microbes consumed. We talk of reducing the BOD, and BOD sewage gets reduced, and the remaining major part of the sludge is pumped into larger tanks. So this is how you need to explain every paragraph. And of course you got a choice. List the events that reduce the BOD, biological oxygen demand, of a primary effluent during sewage treatment. So role of flux and activated sludge in sewage treatment are: flux, these are masses of bacteria held together by slime and fungal filaments to form mesh-like structures. These are used during the secondary sewage treatment in aeration tank to increase the rate of decomposition. The microbes digest a lot of organic matter, converting it into microbial biomass and releasing a lot of minerals. As a result, BOD of the sewage reduces. As the BOD of waste is reduced to 10 to 15% of new sewage, raw sewage, is passed into settling tank. In these tanks, flux are allowed to undergo sedimentation. Now, sludge. The sediment of settling tank is called activated sludge. A part of it is used as inoculum in aeration tanks. The remaining part is passed into larger tank called anaerobic sludge digester. In these tanks, anaerobic microbes are present that digest the organic mass as well as aerobic microbes of activated sludge. The remaining sludge is used as manure. So this is how we are converting all the waste that is being dumped in the cities to useful products. And this is the need of the hour for not